Hello, everyone. Today I'll be talking about the work I've been doing in the lab of the Wang lab for the last 3 years, which is to develop monoclonal antibodies in order to study bat immunology. Okay, the first part, I'll just give a brief introduction on why we want to study bats and delve a little bit more into bat viruses and immunity, and also talk about our captive colony of Eonycteris spelaea bats. Then next, I will delve into the work I've been doing, which is the creation of bat-specific monoclonal antibodies, or MABs for short, and also the application of them, which is the characterization of the bat immune landscape. And finally, I'll end with some of the future research directions that we have been working on. Okay, so why do we study bats? Bats have a lot of unique traits. Some are pretty well known, like for example, bats are the only mammals capable of powered flight. Some of them also have echolocation capabilities, shared with few other mammals. Bats, there are many species, over 1,400 species across the world, and distributed across a very wide geographical range. And therefore, they have a lot of adaptations to the environment, including, for example, cold stress resistance, like hibernation and torpor. And on the flip side, heat stress resistance during the highly metabolic activity of powered flight. Additionally, with their wide range geographical distribution, they also have a wide range of diets, ranging from insectivorous bats to nectarivorous bats, like the cave nectar bat that we work on, and even in some cases, a blood diet like the vampire bats. Bats are also known to have long lifespans relative to their small body sizes, together with cancer resistance. And for today, what I will be mostly focusing on are the fact that bats are a large viral reservoir, and together with a dampened inflammatory response, and we want to understand how these two factors link together. Bats in recent times have become pretty famous for being the reservoir host of many viruses that have, when they cross the species barrier from bats to humans, they cause very severe morbidity and mortality. Like, for example, the coronaviruses that most of us are familiar with by now. Bats can also reservoir hosts for other deadly viruses, like for example, the filoviruses like Ebola and Marburg, and the paramyxoviruses like Hendra and Nipah. So in bats, many of these viruses can be tolerated. In other words, the bats can have infections of these viruses, but they display very few signs of actual viral pathology. Right? But however, when these viruses are very deadly, when they cross the species barrier to humans. And why is this so? Our lab and others have been studying the regulatory mechanisms that contribute to this phenotype, which is termed viral disease tolerance. And generally speaking, there are two main opposing regulatory mechanisms. The first is an elevated baseline antiviral state, where certain genes like your interferons and various antiviral defense pathways are elevated at the baseline level, so that viruses can be controlled at the point of infection. And this is coupled together with a dampened pro-inflammatory response, especially in the NLRP3 inflammasome pathway, where viral infection doesn't trigger a systemic pro-inflammatory response, and together with all the various side effects caused by widespread inflammation. Okay, in our lab, we have access to a captive colony of cave nectar bats, also known as Eonycteris spelaea, similar to how many other labs around the world have mouse colonies. So cave nectar bats are a local species of bat that are found across Southeast Asia. Um, their conservation status is non-threatened, so they are not endangered in any way. They are pretty small, and this colony has been established from about 20 wild-caught bats almost a decade ago, and this colony has been thriving very well. About 300 pups have been born in captivity, and the current population size of this colony is about 160. So what we have done is these bats are housed in a aviary that has been specifically built in order to house these cave nectar bats. So this aviary has a large area in order for free flight and also together with some enrichment to keep them healthy, together with roosting areas that are sheltered from light. These bats are well taken care of with periodic health screening, and we have many studies that are currently ongoing, including ones on immunology, cardiology, aging, metabolism, and heat stress. So basically, just about everything that we can think of to study. A previous publication that studied the bat immune response to viral infection was performed by other members of the lab. And what was done was to take a bat infected with a virus, and 4 days post-infection, perform single-cell RNA sequencing and compared this with uninfected bats. And so this was done in lung samples. And what was found was a whole bunch of different immune cell types that responded to the viral infection. And of course, this is not very surprising. But what was surprising from this data was the identification of two unique immune cell types that express very interesting gene pathways that were not seen in infection in, say, other model organisms like mice, for example. One subset was these myeloid cells that express collagen-like genes and are hypothesized to be involved in tissue repair. And another were neutrophils that were expressing tryptophan degradation genes that are generally known to be immunosuppressive. So currently there are ongoing studies in order to figure out what is going on with these different unique cell subsets and how do they contribute to the antiviral immunity in bats. Okay, so this was one initial study of immunity in bats, and there are several barriers that prevent us from making a lot more studies of immunity in bats. And one of the biggest limitations is that we have very few monoclonal antibodies that are cross-reactive for our E. spelaea bats. So cross-reactivity of antibodies rely on there to be shared epitopes between the original target and our cross-reactive target. And to better illustrate this, I have a diagram here. So for example, this is a protein, a hypothetical protein in mouse and human, where the different epitopes are indicated by the different colors. So generally speaking, for an anti-human antibody raised in mouse, right, there are these shared epitopes. We can have, we can have antibodies against these different colored epitopes. Now, when we introduce bats into the equation, bats have a different set of epitopes compared to mice, human, and bats, right? And if we want to use one of these antibodies made in humans, the only potential cross-reactive antibody or map is at this position over here. And any maps to these other targets will not be viable. However, if we actually make bat-specific maps, right, then now these are the possible epitopes that can actually be targeted if we actually immunize to search for bat-specific epitopes. So to illustrate this problem, thus far, after many attempts, we have only identified about 10 cross-reactive maps so far, which is very few. And the other problem is that these maps are not the most useful in a sense that they target primarily myeloid-specific receptors or intracellular epitopes. So the problem with intracellular epitopes is that you need to permeabilize the cell, so you cannot do any transcriptomic studies, and also you kill the cell. So studying lymphoid cells using these cross-reactive maps is very limited. And therefore, these are one of the reasons why I came up with this project. Okay, so delving into the experimental workflow. Um, so basically, this is what I've been doing in order to generate the maps, and I will go through this step by step. Okay. And so the very first step that we need to do is to generate the recombinant protein of interest. Um, Here is an example of a protein. This is actually um, our spelaea CD4 here. And most of the targets we are interested in are actually transmembrane receptor proteins because we want to generate maps against these immunological receptors. So what we need is to convert these insoluble transmembrane receptors into a soluble form. So what we do is we take this extracellular domain only, cut away the rest of the sequence, and then we add a few other components in order to make it soluble and purifiable, which is namely a IL-2 signal peptide in order for the protein to be secreted for easy purification, and a histag for purification. And so after we generate this recombinant soluble proteins, we can proceed to the next step, which is mouse immunization. And so this is the general protocol that we follow. We do a 3-dose strategy where we immunize the mice 14 days apart with 25 grams of protein together with adjuvant. At day 21, which is 7 days after the second injection, we perform an interim QC by bleeding the mice and checking the plasma for antibody reactivity against the target protein of that we generated in the first step. Then at day 31, which is 3 days after the third injection, we sacrifice the mice in order to harvest antigen-specific B cells. And so what we do is that we sort the B cells for B cells that are IgG positive and that also stain for the recombinant protein that we generated in the first step that are actually stained, conjugated to 2 different fluorophores. So we only sort for cells that actually co-stain for both fluorophores to indicate that these are specifically bound and not the off-target stains that we see, not the off-target, non-specific, singly stained cells. Once we have the sorted antigen-specific cells, we will then proceed to clone the paired B cell receptors from the individual cell. And what we use is the 10x Genomics 5'V2 VDJ single-cell sequencing platform. So this is a very classic single-cell sequencing platform, just that instead of transcriptomics, all we really need is the paired B cell receptor sequences for the heavy and light chain. And so what this process generates is a whole bunch of different clonotypes arranged by the frequency, where we hope at least that the most frequent clonotypes are the antigen-specific B cells that have undergone clonal expansion in response to recognition of the target protein. And so the output of the 10x Genomics pipeline is basically this browser over here, where we have the paired antigen receptor genes. And then we can actually take a look at all the antigen receptor genes here, and we can copy the sequences to feed into the next step, which is basically to process the B cell receptor sequences. So the output is generally something that you see here. So it's almost the full length of the B cell receptor sequence containing a 5' leader peptide. The VDJ region that is the VDJ region that is our region of interest, which is the recombined part, and also the C constant region. So what we do is that we take these sequences and we feed it into a program hosted on the IMGT website called vQuest. And what this does is to identify from the full-length sequences which parts are actually the VDJ sequences, as indicated here in the green color. So we can actually truncate these sequences to remove these other sequences, the leader and the constant region, for cloning purposes. And so we take these sequences to feed them into the next step, where we actually start producing the plasmids and supernatants. And this is all done using the Twist platform. So yeah, shout out to them for hosting me on this talk here. So okay, for this Twist platform, for those of you who have used it, there are many different possible options, and the one that we are interested in is this antibody 1. So what we do is that we feed the truncated VDJ sequences into here, and we have them paired up, and then what this does is that it generates clones. It generates plasmids of these, and then Twist will take these plasmids to express them to generate small scale map production. So for this, over here for the vectors, I have my own custom vectors here, because on the Twist platform, generally they only have human antibody vectors. But if you want to clone, for example, in my case, mouse antibodies, you can create your own custom vectors and upload them onto Twist. And so you can do cloning of your own antibodies in whatever vector or backbone you choose. And so using the platform, you can select whether you want the production to be done in 293 or Trove expression systems, size, or whether you want it purified or not purified. For my purposes, I just do it in 293 cells, a small 1 ml culture for screening purposes, and because I'm screening, I just do it only in the supernatant format. And one of the most useful parts of this Twist platform is actually that not only they give you the supernatants, but they also give you glycerol stocks of all these plasmids. So after you are done screening, you can actually just go back to the plasmids, grow them up, and then you can, in your own lab, mass produce, scale up these plasmids and purify them yourself instead of relying on a separate production run, for example. Okay, so once we have these these map supernatants that we have generated in large scale, then we proceed to perform screening. So for screening, I do multiple ways of test validation of my maps to see whether they bind to my target antigen. And so for the first two ways, we take the original recombinant protein we generated in the first step, and we perform ELISA for conformational binding, or Western blots to test for linear epitopes. And because for our studies, we are primarily interested in identifying flow cytometric antibodies. So of course, flow cytometry is the ultimate goal of these screens. So we want to test for flow cytometry reactivity, as you can see over here, where this antibody is reactive, but this is not. Now that we have our the workflow done, so we've generated a large map panel that is available for immunological studies. So over here were the existing cross-reactive maps that were available. So as you can see, most of them primarily target myeloid lineages. there are a T cell and B cell cross-reactive map, but these are limited to intracellular epitopes, as I previously mentioned, making their usefulness less. And now, over these past few years, we've generated a whole bunch of homemade maps targeting many more targets. So now we have actual T cell targets like CD3, CD4, CD8 for the different T cell subsets. We have a few more myeloid maps like CD14, CD16. For B cells, we have two maps, CD21 and CD45RA, which is actually an isoform of CD45 that is expressed specifically on B cells. And of course, we have CD45 itself as a pan-hematopoietic marker. And finally, we also have NKp46 as an NK cell marker that we have generated more recently. So now we have all these maps, then we need to actually start using these maps in order to perform immune characterization. So as an initial proof of concept, we performed a phenotyping of various tissues of the bat. So this is to show that we can actually use these maps in a useful way, and also in order to perform an initial identification and quantification in various immune compartments to see if we can find anything interesting that we could follow up later. And so for this study, we performed it in 3 adult bats compared to mice, and we performed all these in female animals. We isolated and analyzed these immune compartments. So for the major immune compartments, we have the spleen, lymph nodes, blood, and bone marrow. And we also analyzed some barrier tissue like the lung and intestine that we actually sampled across 4 points to cover the various parts of the intestine. So this is the flow cytometric gating strategy that we perform. I'm using spleen as an example. So we have all the gating performed, the subsequent gating all performed, shown here in the orange gates. And the immune subsets that we actually gate out is shown in red. So we have, like, for example, the granulocytes, which have high site scatter, the CD91 positive monocytes divided into classic and non-classic monocytes based on their CD16 expression. We have CD45RA positive B cells. We have the CD4 and 8 T cells. And also, finally, we have this population of NK-like cells, because at this point in time, we did not have our NKp46 marker ready yet, so we could not use it, and we had to use surrogate markers. Okay, so when we performed the study in the spleen, we observed a few differences in bats compared to mice. One of the big ones is, you can see over here, is that the B cells, there are far less B cells in bats compared to mice. When you look at the ratio of B cells versus CD4 and CD8 cells, we can see that the ratio in mice in red is a lot lower for CD4 and CD8 cells, whereas for bats, the ratio is pretty close to 1, indicating that generally there are just a lot less B cells in bats. The next thing is when we look at the monocytes, we see a lot more monocytes in bats compared to mice. And these monocytes are skewed towards the non-classical lineage generally. And we can see here a better representation. So when we look at the ratio in bats is about 1.5, whereas the ratio in mice is about 2 to 3 times higher. As you can see that the classic monocytes, they form a much bigger population compared to bats. And the observations we see in the spleen is not just limited to the spleen, but we can detect it elsewhere in the blood, the bone marrow, and the lung. So for the B cells, we can see once again that there are fewer B cells across all these compartments. And for the monocytes, except for classical monocytes in the lung, we generally see fewer monocytes across all the immune compartments we tested. And additionally, the ratio of classical to non-classical monocytes is more skewed towards classic monocytes in mice, but are more balanced with a lot more non-classical monocytes in the bats. Um, Next, an interesting observation we saw in the bats' splenic CD8 cells was that there were a large fraction of them that express the receptor CD11B, which is generally known to be a myeloid receptor. So you can see over here in bats in black, we see pretty high levels of CD11B expression, but in mice, we do not see it. So it was pretty interesting for us that we um, not just in this experiment, but in other experiments, we consistently saw CD11B expression in our CD8 T cells, and we were wondering what's going on. So when we looked back at, we tried to search for studies on this, and what we found was CD11B, old papers suggesting that CD11B is a marker of activated T cells in humans and mice. And so we are planning to look into this further to characterize these cells to see what are they doing in our bats, since this is meant to be a steady state uninfected bat. Next, we analyze the intestine, and due to experimental issues, we failed to obtain enough cells from the mice, so we could not conduct a comparison of bats and mice. But anyway, for the bats, this is the general structure of the intestinal tract. Um, Bat intestinal tracts are actually very homogeneous in their outward appearance. So we cannot actually tell what parts of the bat these are until we actually perform histological sectioning. So this very rough sectioning is based on histological sectioning of other bats. So what we did is that we took four different sections distributed across the intestinal tract. So one around the duodenum and jejunum area, two in the ileum, and one in the colon and rectum. And so we also, due to very high levels of background noise, we restricted our analysis to only the adaptive immune cells, so the B cells and the CD4, CD8 T cells. And initially, one of the most um, obvious observations is that there are very heterogeneous levels of all cells, of all these cell types. And intriguingly, there were very few B cells in the stomach proximal part and the anal proximal part of the intestinal tract. And so this is interesting because what we expected was that there would be B cells everywhere. And so there could be two possibilities. The first is that simply there are a lot fewer B cells, or secondly, is that plasma cells in general don't express many surface markers. And so one possibility is that there could be a lot of plasma cells and they simply did not show up in flow simply because they don't express the markers that we were searching for. And so in order to look at this in greater detail, we performed histology. So we took the whole intestine of other bats and performed Swiss roll sectioning in order to see the whole intestine. And what we could see is the presence of multiple lymphoid follicles scattered throughout the intestine, as you can see in these light blue arrows, and these are primarily in the ileum. And when we actually zoom in on higher magnification using either H&E staining or CD45 immunohistochemistry, we can actually see the classic lymphoid follicle structure with very obvious light and dark zones that we can actually see, right? So then when we looked throughout the laminar propria, in every single part of the intestinal tract. When we zoomed in at higher magnification, we can actually see these cells indicated by these white stars, which have very characteristic morphology of plasma cells. And since they were found everywhere throughout the intestine, um, our conclusion was that actually there are many plasma cells to secrete antibody throughout the intestine, and our finding of not having B cells in the proximal and distal parts of the intestinal tract is simply because that these plasma cells did not show up, and the B cells we actually detected were the ones that were present in these lymphoid follicles. So unfortunately, we did not find out, we did not make any big amazing discoveries, but sometimes that happens. Okay, so moving on from this study, we have a few future research directions planned. Um, the first is that we've not only made antibodies for immunological receptors, we've actually made antibodies that are specific to different antibody isotypes. So we have IgG, IgM, and IgA for our E. spelaea bats. And for IgG and IgM, we tested their cross-reactivity to different species of bats. And special thanks to the lab of Gavin Smith for providing many of these samples. And for IgG and especially for IgG and to a lesser extent IgM, we saw cross-reactivity of our maps to many of these species. And so what this means is that the tools we actually generated here are not just limited for use in our colony, but could also potentially benefit the broader community out there if the organism they use has cross-reactivity to these maps that we generated. Another future research direction we are working on is to perform TCR sequencing. So using a process of BLAST and also some manual annotations, we've basically generated annotations for the V, D, J, and C gene segments for TCR alpha and TCR beta, where you can see here for TCR alpha, there are 77 V gene segments and 55 J gene segments, whereas for TCR beta, there are 57 V gene segments with 1 and 57 over here, 2 Ds and 12 J gene segments. And so these annotations were made possible by a combination of homology searching and together with um, refining by performing 5' base sequencing of bulk splenocytes and using desequencing in order to identify and annotate the various gene segments and pseudogenes. And you can see example output here of TCR beta usage, VJ gene combination, gene segment usage in the spleen. And as you can see here, there is a pretty diverse usage of the various V and J gene segments, with some that appear to be used less, and this is pretty expected. So, what we want to do in the future is essentially to optimize this assay in order not just for use in single cell, or sorry, not just for use in bulk cells, but also to actually transfer this assay in order to perform this in a single cell transcriptomic assay. And currently, work is undergoing in order to optimize this. Okay, so this is pretty much my talk. I would like to acknowledge the many people that are involved in this work. So this is my lab. We are a massive lab with a lot of people, and many people contributed to this work. Of course, my PI Linfa over here. Without him, none of this could have been possible. Many people have contributed, like for example, Randy, who is the bat colony manager. Many people have helped with the bioinformatics works, and together with other aspects like the mouse work and whatnot. And the whole list is here. There's too many names for me to mention, and together with anyone else who have helped me else. We have also a research collaboration agreement with Paratus Sciences, who have provided some of the funding and also assistance in creating these maps, together with NLAF for husbandry and care of the bats themselves. And so I also like to acknowledge some of my funding sources, the NRF and also Paratus Sciences. Okay, so now I can take questions, and I'm also contactable at this email address here if anyone wants to send any further questions, or if, for example, you want to perform reagent sharing or possible future collaborations. So yeah, thank you so much.